Okay, everybody, on a scale of 1 to 5, 1 being amazing, 5 being there's room for improvement, how would you rank your week? I think I'd put myself at like a threes and fours. I think I'd go like a 2 or 3. You know what I'm thankful for is that nobody did a 2.5. Now somebody's gonna do like a 3.5 or 1.1 or something like that. Nobody did. Okay. Um, so let's hope that after today's class, I can do my part to maybe make your week a little bit better. Um, what we are talking about today is we're going back to that day 6 protein structure 3 and 4 slide deck, and it is the one that starts like the screen that you're about to see. And I also want to the bad habit of opening a file and then not closing it. Okay, now that's open. Now I'm going to share my screen and share the right screen or the correct screen. Here we go. It's this slide deck. I'm trying to use the phrase slide deck instead of PowerPoint because I think that PowerPoint is just super redundant. But it's something that I almost always do. And I have the bad habit of naming a PowerPoint file presentation 1, presentation 2, or new presentation. So, anywho, here we are. What we're talking about is proteins and 3D structure. The other day on Tuesday, what we talked about was we predominantly spoke about fibrous proteins. So we talked about sarcomeres, which are the individual units that make up muscles. And then we talked about the proteins that are involved in those. We also discussed how those sarcomeres change. For instance, what happens between a myosin head group and an actin filament? What inspires or causes an actin filament to be released from a myosin head group? And that was the binding of adenosine triphosphate or ATP, which is a major and super important molecule when it comes to um, energy and energy availability for basically every organism. Then that's your energy currency. Then we talked about globular proteins. And this is kind of where we took off, and we're going to jump back into this. Globular proteins, they have an unfolded state and a folded state. A folded state is their functional state where they're actually going to be able to catalyze and carry out any sort of function and reaction, whereas an unfolded state, well, that doesn't really carry out any sort of function. In addition to that, notice with this one, there's no like organization, there's no structural elements to it. Whereas in our folded state, this is also known as a native state. That's where you actually see secondary structural elements like alpha helices and beta sheets. And then, in addition to that, random coils. So RC for random coils, and then alpha helix. Okay, can you give me a 1 to 5? 1 being, I've got it, and a 5 being, ooh, still working on it with respect to secondary structure. And yeah, okay, cool. Oh, someone threw a 2.5. Okay, I respect that. That's fine. That's fine. Okay, so our folded and our unfolded states. We want proteins to be properly folded because then they can carry out their function. Now, this is the first time where I want to draw a connection between structure and function. Now, there's going to be another element that we're going to add to this later today that, well, it's going to make this almost a triangle of ideas. So right now we have a connection between structure and function. And you know what? I'm going to draw some kind of equilibrium arrows to show that there's a relationship between the structure of a molecule and the function of a molecule. In this case, whenever I say molecule, I'm referencing a protein. There's also a relationship between the function and the structure. Now, we can denature proteins, and one way that we do that is we heat them up. So we take them from this folded to the unfolded state by heating it up. And what we're doing when we're heating it up is we're breaking lots of hydrogen bonds. Hydrogen bonds are fairly weak, but there's lots of them that hold protein structures together. This is an important and powerful slide because it brings to the forefront interactions that exist in proteins. We've got secondary structural elements, and those are going to be stabilized by backbone hydrogen bonds. Now, just as backbone hydrogen bonds stabilize the secondary structure, hydrophobic interactions between R groups, disulfide bonds, ionic bonds, and, well, R group H bonds stabilize the tertiary structure of a protein. Now, your R group interactions. Well, hydrophobic interactions are going to be kind of found at the center or the core of a protein. Hydrogen bonds stabilize polar regions. Ionic bonds, disulfide bonds, and van der Waals forces are also going to be involved in stabilizing these interactions and stabilizing a protein structure. Now, this is what we got to and kind of where we jumped off with on Friday, Thursday, Tuesday. Today is Thursday. Sorry, everyone. Denaturants. Now, these are chemicals. So if you add these chemicals, most of these are chemicals. If you add or if you have a chemical treatment or a physical treatment added to a protein, for instance, a substance known as beta mercaptoethanol or dithiothrietol, what these are going to do is they are going to break disulfide bonds. Now, with that said, disulfide bonds, these are a covalent bond. And these, if you change the pH, I'm going to put a delta pH. They don't care. I'm going to add this little star here and say, don't care. Mercaptoethanol and dithiothrietol, or beta mercaptoethanol and dithiothrietol, these are two different treatments, two different chemicals that are involved in the redox reaction that break and form disulfide bonds. Detergents. Now, I know that whenever I took organic chemistry, we talked about detergents. Detergents are chemicals that will disrupt hydrophobic interactions. So think about your alkyl functional groups. So like if you have an interaction between ILE or isoleucine and leucine, these two amino acid R groups are interacting because they're both nonpolar. They both hate water, so they're trying to get away from it. A detergent will disrupt that interaction. If you do a heat treatment of a protein, what you're aiming to do is you're aiming to break a bunch of hydrogen bonds. So amino acids like serine and threonine, for example. Add enough heat, the interaction between those two R groups will be disrupted. Alter the pH, and you're going to change charge of different amino acid R groups, and therefore you're going to be altering ionic interactions. So D for aspartic acid and R for arginine. Well, if you adjust the pH super low, well, aspartic acid isn't going to be deprotonated, and therefore it's not going to have a negative charge. So you're not going to have those charges that kind of stabilize an interaction. Chelators, these bind metal ions, and well, metal ions are some ways in which different amino acid R groups will interact with one another. Now, urea and guanidinium chloride will break hydrogen bonds. These two compounds are known as chaotropes or a chaotropic agent. So they really love disrupting hydrogen bonds. Now, one question for you. Thinking about this one at the top. If you remember, I said that if you change the pH, a disulfide bond doesn't care. Okay. Now, with that in mind, do you think that the addition of something like urea will alter or will disrupt a disulfide bond? At a 1 if you think that it will, a 2 if you think that no, it won't. I like it. I'm seeing lots of 2s, and you're absolutely right. Now, this to me, one of the things that's important about it is to think, man, hydrogen bonds. What, like, this is going back to like Gen Chem 1 and Gen Chem 2. You learn about hydrogen bonds in Gen Chem 1, and then you go deeper into them in Gen Chem 2. Hydrogen bonding is one of the forces that really makes water unique. It's what helps water have high surface tension. It's what helps water have a high boiling point and a low melting point. So these physical property or these physical properties of water are severely influenced by hydrogen bonding. To disrupt hydrogen bonding in an aqueous environment, that seems to me almost to be like a really dramatic alteration. And it is. If you disrupt hydrogen bonding, then man, a protein structure is just going to fall apart. But if it has disulfide bonds, those disulfide bonds will remain intact because disulfide bonds don't care for or about beta mercaptoethanol and dithiothiazole. Can you give me a Y if you follow that and an X if, wait a minute, that doesn't make sense. And if you got an X for that, please follow up with a question like, what about that does not make sense? Okay, I'm seeing lots of Y's, so I'm going to proceed. Now, this is an important concept. What I was just talking about of this, the addition of urea throws off hydrogen bonding. Now, here on the left-hand side, we've got this protein known as RNase. This is kind of an artist rendering of the native structure of ribonuclease. Ribonuclease is a protein that sees RNA molecules and just chomps them up. Chomp, 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 chomp. Now, there's tons of different RNases. So we're just going to go with this one. This specific one is RNase A. Okay. Now, this was studied and has been studied and continues to be studied um, because it was a great model. It was easily purified. And whenever I say easily, I mean relatively easily purified. And this fellow right here on the right-hand side, his name is Christian Anfinsen. Now, Christian Anfinsen, I believe he was at Iowa State University. I can't really remember with a huge amount of certainty, but I believe it was at Iowa State. What he studied was, well, he got RNase and he said, all right, well, I know that if I have native ribonuclease, it will function. And I know exactly what it will do. I'll give it RNA. So here's my RNA. I'm going to give it RNA, and then I'm going to give it chunks. It's going to spit out chunks. These X's represent chunks of this RNA molecule. Can you give me a one if you follow that? Okay. Now, what Christian Anfinsen said was, this is great. I understand this. Nobody's really doubting this. I've got this way to study this enzyme. Great. What I want to do, though, is I'm going to study the structure of this protein. And what I'm going to do is I'm going to add urea. 8M urea. Could anyone add to the chat what that 8M stands for? 8 molar. Perfect. So I'm going to get a solution and I'm going to make in my reaction vessel, I'm going to get 8 molar urea. Super high concentration. What does urea do? It's going to bust up all of these hydrogen bonds. So he says, I'm going to get 8 molar urea, and then I'm going to add some beta mercaptoethanol. And I'm going to add the beta mercaptoethanol because I know that here, 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 and here, there's a couple of different disulfide bonds. If I don't add the 8 molar urea, those bonds will remain intact. Or sorry, if I don't add the beta mercaptoethanol, those bonds will remain intact. So I'm going to add the beta mercaptoethanol. I'm going to add the urea to bust up all the hydrogen bonds. I'm going to add the beta mercaptoethanol to bust up all of the disulfide bonds. And whenever I do that, oh, I'm going to end up with this right here. All of my disulfide bonds have been reduced. So I no longer have those disulfide linkages. I've got a bunch of free cysteine residues. It's denatured. I fully denatured this protein. And now if I give this RNA basically the reactant that it needs, well, nothing happens. No reaction takes place because, well, this protein has been denatured. Now, to that point, everyone was like, Christian, we know. And he was like, yeah, I know that you know this, but here's where it gets tricky. I know what happens whenever I add the 8 molar urea and the beta mercaptoethanol, but what I want to do is I want to try to remove the 8 molar urea and the beta mercaptoethanol. And at that point, everyone standing around, this is back in the 1950s and 60s, and they went like, they did that number. Every single monocle fell out of everyone's eyes. Monocles were shattered everywhere. It's the 50s and 60s. I've decided that people wore monocles. Everyone was just like, Christian, what are you going to do? Why? He was like, the reason I'm going to do this is, hear me out now. If we remove the urea, do you think the hydrogen bonds will go back intact and those will no longer be disrupted? And everyone was like, yeah. And then he's like, what about if we remove the beta mercaptoethanol? Will our disulfide bonds form again? And they say, I, I guess. And he's like, okay, so hear me out. What I think is if I remove this stuff, I can go from my fully denatured form of my protein back, back to the native ribonuclease. And whenever I go back to the native ribonuclease, it will be functional. And everyone, again, they had replaced their monocles, new monocles fall, and they shatter everywhere. Are you serious? And he's like, yes, I'm serious. And they're like, okay, well, next lab meeting, you better be ready. And you know what? Next lab meeting, Christian Anfinson showed up. And he said, guess what, guys? Whenever I do the experiment that I said I was going to do, I took my denatured protein and I renatured it. And everyone was like, that's great. From a technical standpoint, that's cool. But even bigger than that, here is the big thing. This is the, actually, here, I'm going to use the type tool. Let's see. Eraser, eraser function, not to be mistaken with the Arnold Schwarzenegger movie, Eraser. If anyone's ever seen that, it is a movie. The big deal. I'm going to do all caps. The big deal. Think about the different structures. Think about the different levels of protein structure. Primary structure is all you have when dealing with a denatured protein. Okay. C-ANF. I can go with C-ANFs. Is everyone good with that being our nickname for him? Give me a 5 if you're like, okay, I follow. Okay. C-ANF's conclusion is to go from primary structure to tertiary structure. There's no special sauce. There's no magic. A protein will naturally fold into the native state. Now, can you give me a 7 if you follow that statement and that idea? Okay, great. Now, on one hand, you might look at that and be like, oh, okay, that makes sense. Got it. Now let's rewind for a second. What did we need to know in order to predict secondary structure of a protein? Well, we had to know the primary structure of a protein. And so it was like, okay, well, you can use primary structure to predict secondary structure. You can make a reasonable estimation if there's an alpha helix or a beta sheet or or if there's not in certain locations. Give me a 9 if you follow that idea. Okay. Now, here, what Christian Anfinson's work proved or theorized, I'll say, was that, hey, you can use primary structure to predict tertiary structure. Now, that's a big deal because when you look at a protein, and I'm going to close this off, and when you look at a protein structure, this one on the right hand side, there's some random coils. You can't predict the random coiling. You can only, like with the, sorry, when you're predicting secondary structure, you can predict, hey, there's a likelihood that there's an alpha helix or a beta sheet here. The random coils, though, all you can do is say, well, it looks like there's not an alpha helix or a beta sheet here. So Christian Anfinson's work basically was that is a part of kind of that next step of, hey, we can use the primary structure of a protein to predict the tertiary structure. And that's huge when it comes to protein structure. Now, what like the next step of this and the next important point of this is that, well, if you know what a protein looks like, if you know what a viral protein looks like, if you know what any sort of bacterial protein looks like, you now have insight into, hey, that's what this thing looks like on the molecular level. You know what I'm going to do? And I always draw enzymes and proteins to basically look like Pac-Man. Hey, this is what my protein looks like. Oh, you know what? I'm going to design a small molecule that fits right here. And whenever that small molecule fits right there, well, this thing right here is not going to function. You can kind of build like a molecular wrench that you put in the spokes of this protein's function. Can you give me a 3 if you follow all of that? Okay. Okay. So we are going to proceed from here. Important terms, I kind of touched on these, or I've kind of gone over these, so there's a little bit of a review. But a native protein structure is the state that a protein is in when it can function. Think about this as its working shape or fold. All bonds and interactions are intact. Denatured, well, it's the opposite of that. It's no longer functional. And bonds and interactions are disrupted. What bonds and interactions? Disulfide bonds, hydrogen bonds, ionic interactions are disrupted. So all of those covalent and non-covalent interactions are disrupted. Now, with that in mind, what does a protein do? Well, and how is a protein made up? Sure, we can look at these neat little ribbon diagrams and say, oh, well, that's what this protein looks like. But there are some things that are not quite that simple. So, for example, there are domains on a protein. What is a domain? A domain is a region of a protein that folds independently that has a specific function. So, what I mean by that is, here's the protein's whole structure, but this part right here, you know what? I'm going to change colors for that. This part of the protein, what that does is it makes its own little shape, and this carries out a specific reaction. Okay. So what that means and what's important about this is if I were to get some sort of molecular scissors and cut this right here and take this chunk and remove it, that small fraction would do its own thing. It would carry out that same reaction. It would fold into the right shape and it would clip, clip, clip. Okay. Or it would carry out whatever reaction it does when it's a part of a protein as well. So it's almost like is anyone familiar with the add a Y or an N to the chat on this one? Has anyone ever heard of grafting plants or trees? So that's where you get like and you have to do it. There's a level of relationship that they have to have. But think about if you had an apple tree, or actually we'll make them both citrus. Let's say that you had a lemon tree in your backyard, and then your neighbor had an orange tree. If you went and cut off a branch from that orange tree, and here's my, what did I say? Lemons, lemon, tree. That's a lemon tree. Okay, it's gotten worse. And then I'm going to switch colors to give it an orange color. An orange tree, what you're going to do is you're going to basically make a small incision in this tree, and then you're going to put a branch from an orange tree on it. Well, this tree will still grow, and you will have a tree that produces both lemons and oranges. Um, so this part would be like a separate orange domain. Can you give me a Y or an N if that makes sense? Okay, great. Great. Um, Now, that's kind of a parallel to a protein because this whole tree would be the protein, but this part would have been like a specific domain. Now, you might be thinking, well, is that something that happens like within a cell? No, it's not. It's something that has happened evolutionarily over millions and millions of years. Now, characteristics of a domain. A domain can evolve, function, and exist independently. So we could cut this, we could take this protein and cut this off. I'm going to erase the tree that I drew a moment ago, despite it being like a really good drawing. We could cut that part of this protein off and it's going to be able to do its own thing. Now, a domain is typically 25 to 500 amino acids. It's also going to be found in multiple proteins across different organisms. So this same domain is going to be what's known as highly conserved. It's found in this protein, it's found in 50 other proteins. Now there are types of domains. One type is a catalytic domain. A catalytic domain does a specific reaction. So it performs a catalytic activity. So what that means is that it breaks a bond or it makes a bond. Maybe it adds a methyl group, adds methyl group to a molecule or to another molecule. A binding domain. Well, a binding domain simply binds to DNA, RNA, or other proteins. A structural domain is simply going to contribute to the overall protein stability. So it's just basically there to like hold everything intact. Um, it's almost like yeah, well, so we have three types of domains: catalytic, binding, and structural. Structural, it gives the protein a pose. I wasn't intending to be like, hey, your camera froze, but I also kind of, that's fun to do sometimes. Catalytic domain, it does something. My hand is a catalytic domain. It's snapping, it's catalyzing a reaction. A binding domain, my left hand now is a binding domain because it grips and holds on to my Diet Dr. Pepper and just holds it. Okay. Now, as an example of this type of protein, this is a protein on the left-hand side that I don't need you to necessarily know what this does, but I want you to know, or I want you to like use this and plant the seed for this, or take this and plant it as a seed. GAPDH. Okay. Now, GAPDH stands for glyceraldehyde 3-phosphate dehydrogenase. I'll just give you the important context. This is an important enzyme in sugar metabolism. Okay, so it's an important enzyme in breaking down sugars. It has two major domains. One of them is a binding domain. The other is a catalytic domain. Now, the binding domain binds onto the molecule that you're going to be acting on. It's almost like it picks up this sugar related molecule so that I can then hold it in place so the other domain can act on it and carry out an oxidation reduction reaction. So, what I want you to think about with respect to this is if we were to basically cut this in half, well, the binding domain is going to do its thing. It's going to bind onto the molecule that it needs to bind onto. But you know what? Whenever it binds it, it's just going to hold it. In the absence of the catalytic domain, which then goes on to like break a bond or catalyze some reaction like that, well, think about it like this. Let's say that okay, my right hand is going to be my binding domain for my Diet Dr. Pepper. My head, including my mouth, is going to be my catalytic domain, which is responsible for drinking the Diet Dr. Pepper. Let's say that my hand is right next to my head. Okay. When they are right next to each other, when they are in close proximity, does everyone follow? When they're in close proximity, they can work together. And my hand will hold on to this Diet Dr. Pepper can until there's no Diet Dr. Pepper in. And so let's say that I chugged the Diet Dr. Pepper. My right hand, my binding domain would be like, oh, it's empty now. So whenever it's empty, I'm going to release it. Give me a 3 if that makes sense. Okay. So now just imagine if my right hand is on the other side of the room. 3.14. Nice. I like it. My right hand's on the other side of the room. Will it bind a full Diet Dr. Pepper? Yes, no doubt about it. Well, my right hand is super far from my head. So is my catalytic domain going to be able to do anything? No. So that's the first part. The second part, if my right hand is holding on to this Diet Dr. Pepper, is that Diet Dr. Pepper ever emptying? No. So my binding domain is going to grab onto that and hold on to it indefinitely. It's only when they're right next to each other that they can kind of work together to complete that function. Now, this is in an aqueous environment, so if the domains are separate, will they ever come in contact? Yeah, possibly. But the rate of that reaction is just going to plummet. Now, what I want you to take away from this is binding domains have a designated and specific function. Binding domains will bind one specific molecule. Catalytic domain will act on one specific molecule. If the binding domain, if you, let's say that a binding domain were able to bind to a methyl group, that's a carbon with 4 hydrogens surrounding it. Could it also bind to dichloromethane? Nope, they're different. So it's not going to bind that. Now, when it comes to proteins and the processing of proteins, chaperonins are classes of proteins that basically help a protein fold properly. One example of a chaperonin is known as the GRO-EL, GRO-ES complex. Sometimes just because my name is Joel and I pronounce it Joel, I would read this as a graduate student. No, Grohl. Not the case. That's not the case. It's called GRO-EL or GRO-EL, GRO-ES. So this protein complex, I want you to think about this as a good example of a quaternary structure. So there's a bunch of different polypeptides. Now, when a misfolded, mis or partially folded protein is present, well, that partially or misfolded protein will go into the central cavity of this. And this GRO-EL, GRO-ES complex gives a really unique environment for that protein to be refolded. Okay, so misfolded or partially folded protein goes in, and a properly folded protein goes out. Properly folded goes out. Now, do all proteins need to go through this GRO-EL, GRO-ES complex? No, they don't. Some do, but not all of them. So proteins can be processed if they are mis or partially folded with the support of a chaperone. In addition to that, there are proteins known as proteasomes. Now, a proteasome is a protein complex that degrades proteins. It takes a, excuse me, I apologize. It takes a protein, and this could be a properly folded protein, a misfolded protein. I'm just going to leave it a general protein. And what it will do is it will put it into, I always kind of think about this as like a trash compactor. It takes a protein, puts it in here, and dices it up into 8 amino acid long peptides. So what I mean by peptides that are 8 amino acids long. So if you have a protein that is 275 amino acids, well, that's going to break it up into somewhere in the order of, let's see, that would be like 33, no, 34. Yeah, 34 peptides. This is not a point where the math is important. Okay. Don't think like, oh, but is it actually 8 point blah, blah. No, don't worry about that. Just breaks them up into these shorter peptides. Now, how does that happen? It helps to have this system known as ubiquitination, or this process. So proteins don't randomly show up to a proteasome and get digested. Instead, what happens is they get this protein right here added to them. This protein is a small, I think it's 76 amino acids long. It's a small protein known as ubiquitin. And ubiquitin is added to a target protein to the point where it looks like this. This substrate right here, or target protein, is the one that you want to degrade. Okay. So what you're going to do is you're going to put a total of at least 4 ubiquitin subunits onto that protein. And when a protein has 4 ubiquitin subunits covalently bound to it, then it goes to the proteasome. So you could think about this almost as if a protein gets ubiquitinated, chances are it's going to be degraded. If a protein does not get ubiquitinated, does not have these ubiquitin subunits added to it, it's probably not going to be degraded. Okay. So what I want your brain to think, whenever you hear ubiquitin, think, aha, that's the protein that helps target other proteins for digestion. Can you give me add an emoji to the chat if that makes sense? Okay. Okay. If you did not put an emoji there, follow up with a question as to what does not make sense about this. But what I want you to take away from this, are you saying turns into a proteasome? No, I'm not. The proteasome is your protein that degrades other proteins. So specifically, the proteasome is the protein complex that degrades ubiquitinated proteins. Is ubiquitination only for digestion? That is an amazing question. And I would say, no, it's not. The reason that I would say no, it's not is because, well, it's not. Um, but also because of the fact that sometimes, like if you get this chain right here of 4 ubiquitin subunits, then chances are that protein's going to get degraded. So I'm going to put a red star or a black star over substrate. That's our protein that's going to get degraded when you have these four ubiquitins. Sometimes proteins get one ubiquitin added to them. Sometimes they get two. They don't get four, chances are slim that they're going to be degraded. So that's why I say it's not only for digestion. Can you explain the notation you made with an arrow to proteasome? I'm saying that this, what I put in a big circle here, goes to the proteasome. When you've got four of them. So those four ubiquitin subunits mark it for degradation. When it's just one, it's not necessarily going to do it. And I want you to operate under the idea if it's only got one ubiquitin, it's not going to get degraded. Okay, but you just want us to know it helps with digestion. Yep, because this is a first semester biochemistry class. If you pursue this further, you'll learn all the nuances and subtleties of this. But for the time being, what I want you to know is that this right here. Okay, we got two slides on this slide deck. Then we're going to talk about a particular protein and its function, or a pair of proteins and their function. IDPs. IDP stands for intrinsically disordered proteins. Okay, so what that means is that, well, it's got some really crazy shapes to it. An IDP is a protein with a bunch of wild shapes. Now, not all proteins are folded into stable structures. So, what that means is that you've got a lot of mobility with an intrinsically disordered protein. IDPs have regions that favor disorder, that favor not really having. It's almost like the random coil is super random. An IDP region tends to lack hydrophobic residues. So IDPs don't have many hydrophobic residues, which you could also know as your nonpolar residues. They are rich in polar amino acids, but there's one stickler, proline. Proline is commonly found in intrinsically disordered proteins. IDPs may favor adaptation to binding another protein. They may favor being modified. They may be more involved in signaling and regulation. And non-IDPs are involved in catalysis and transport. So these proteins, they have this, it's almost like, um, I always think about an intrinsically disordered protein as similar to like I'm sure there's so many different examples in like fiction or something like that, but one of the things that has drawn people to the Mona Lisa, and not an art critic or art commentator, but this is the closest that I got, is when you look at the Mona Lisa from different angles, it gives you a different like perspective and vantage point. No, here from this angle, it looks like the the person is smiling. From this angle, it looks like not a smile. From this angle, it's this. It's different from so many different angles. An IDP is kind of similar to that. If you look at an IDP and then you look at it from a different angle, or if you give it more time, it's going to change its shape. It's very, very dynamic, more dynamic than your typical protein that has like clear secondary structural elements. Okay. So this is the bulk of information that I want you to know about intrinsically disordered proteins. Proline and other polar amino acids are pretty common. Now your quaternary structure, this is our jump off to our next slide deck because of the fact that, well, this is going to be our foray or our step into protein structure and protein function. Now, when you talk about quaternary structure, on the right-hand side, we see the protein hemoglobin. Hemoglobin has four subunits. Within each one of those subunits is a heme ring. Right there. Oh, dang it, that's not what I wanted. Um, doop. Right here, 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 and here. Hemoglobin is a protein, or HB as it's commonly known, is a protein that transports oxygen. Now, this with four subunits is known as a tetramer. Structurally, the interactions between these subunits are the same as what we see between R groups on a single monomer. So what that means is that there's ionic interactions that stabilize this purple one and this green one. There's hydrogen interactions between the olive and blue one, and then the green and the blue one. There's also hydrogen bonds and ionic interactions, all the above. There's also going to be hydrophobic interactions. Now, the hydrophobic effect is this idea that, well, hydrophobic or nonpolar groups are going to cluster together to get away from water in an aqueous environment. Aliphatic residues are going to repel water and they're going to be driven together. So when it comes to the quaternary structure, yeah, there's hydrophobic residues that interact closely with one another to kind of keep that water away. An example protein for quaternary structure is hemoglobin, which is what we see right here. Now, I'm going to switch my share to our next slide deck, which the very first slide reads this, protein function. I'm going to jump right into this because, well, hemoglobin is a model that we use for the study of quaternary structure, and it's a model that we study for protein function. So when we study, did I not add it to the calendar? Joel, man, pause. Dang it, dang it, dang it. I'm sorry, everyone. Thank you, Dylan. Thank you. I didn't. The good thing is that we have high-speed internet connections as opposed to like a dial-up modem. It would have been kind of funny if we had a dial-up modem, because then it'd be like, hey guys, in 12 minutes, actually no. When I was in when I was younger, I'll just leave it at younger. I remember my friend downloaded the song Stairway to Heaven by Led Zeppelin, and it took an hour and 20 minutes. And it's the sort of thing that I say, and I'm like, wow, I feel like a dinosaur. That's right. I feel like a dinosaur. Awesome. With the ability to eat an entire goat in one sitting and to rip down electrified fences like in the movie Jurassic Park. Okay. Slide deck is up. I will post the PDF a little bit later. Sorry about that, everybody. Here we are. Protein ligand binding. The function of many proteins involves the reversible binding of other molecules. This molecule is a ligand. Okay, so my Diet Dr. Pepper can is my ligand. My protein is my left hand. Now, ligands, the molecules that are bound, actually, you know what I'm going to do? I'm going to add the link to our slide deck to the chat. There we go. Okay. Um, Ligands can be a variety of different molecules. They themselves can be proteins, they can be small organic or inorganic compounds. Ligand binding, importantly, is non-catalytic, often uses a protein conformational change, so a shape change, and it can be regulatory. Okay, now I bring all this up because one of the important distinctions is this first item. Ligand binding is non-catalytic. So what that means is whenever somebody uses the terms protein ligand or protein and ligand, what they're meaning is that this protein will bind to a molecule and then release it. It will not change it. This is different and distinct from an enzyme. An enzyme binds to a molecule, but it doesn't, like the language, that is not a ligand that it's binding. That is In that case, we're talking about a substrate. So a protein binds a ligand, releases it, binds, releases, binds, releases. When it comes to or enzymes, you're binding and you're breaking a bond and you're releasing it. So ligand binding is non-catalytic, often uses a protein's conformational change, and it can be regulatory. So that regulatory instance is, well, we're slowing a process down or we're speeding a process up. Oxygen transport, this is kind of our overview of what's taking place. In the lungs, we've got a high concentration of oxygen. In the tissues, in our muscle tissues, we have a low concentration of oxygen. But you know what? My muscle cells, just like everyone else's, we need them to generate a bunch of ATP because, and just think about the connection. When we break sugars down, break down here, or broken down here in our tissues, we get a bunch of ATP. Have we talked about ATP helping muscles move? Yeah, we have. ATP is essential for myosin head groups to release from an actin filament. So we want this to happen. Now, the way that we can get a bunch of energy, a bunch of ATP from a sugar, is by using oxygen in aerobic metabolism. There's anaerobic and there's aerobic metabolism. Anaerobic means no oxygen is utilized, whereas aerobic metabolism is that which uses O2. So we've got to get oxygen from our lungs to our tissues. How do we do that? Well, there's a protein known as hemoglobin, which I will denote as HB. And what hemoglobin's going to do, and this is kind of tough to see, but hemoglobin's going to pick up one, two, three, four O2 molecules from the lungs, and it's going to transport it through the bloodstream until that fully oxygenated or oxyhemoglobin gets to the tissue where the O2 concentration is low. Well, that oxyhemoglobin will release O2. That O2 can go to this structure. Can anyone add to the chat what that structure is? You know what's awesome is I've got four people who have typed mitochondria. I bet there's, let's see, we've got attendance of 41 today. I bet there's at least 10 people who did this, or mitochondria, or typed it or something. But yeah, you're absolutely right. That's the mitochondria. That mitochondria, that structure right there, is going to generate a huge amount of ATP with the help of this O2. So O2 getting to the mitochondria within muscle cells and within other cells is very helpful. Now, there's another protein within those tissues known as myoglobin. And myoglobin within the tissue is just going to hold on to O2. It's almost like oxyhemoglobin delivers a lot of oxygen, an excess or a surplus of oxygen to the tissues. Myoglobin holds on to excess O2, and then whenever it's needed, it will release it. Now, the byproduct, or one of the products of sugar metabolism, is carbon dioxide. Well, that carbon dioxide in the tissues isn't where we want it. So we've got to get it back into the lungs so it can be released. And so we can exhale and get rid of that CO2. Now, hemoglobin's going to help out with that as well, and we'll get into the details of that a little bit later. But primarily, what hemoglobin is doing is taking oxygen collected in the alveoli, right here in the lungs, delivering them to delivering the O2 to the tissue, so we can go through sugar metabolism. Now, let's talk about these two proteins, myoglobin and hemoglobin. Myoglobin functions as an oxygen storage and transport protein in the muscles. Okay, so that's myoglobin. Myoglobin is the player that holds on to O2 in the tissues and just does that. Holds on to it, then when needed, releases it. Hemoglobin does the same thing, but it is not stuck in the tissues. it is in the blood vessels. So it's going to be mobile. Hemoglobin from the lungs goes all the way to the tissues and releases O2. Myoglobin and hemoglobin are built on a common structural motif, ooh, also known as a globin fold. Now, this is similar to a domain, but a motif is something that you're going to see in its sequence, primary sequence. Now, what I want you to know about this is that basically, if we look at myoglobin right here, it is a monomer. Now, give me a 1 if you can see the letter A that I've just circled. Okay, great. Now, this A, what it corresponds to is that is an alpha helix. Hemoglobin and myoglobin are both a bunch of alpha helices. A, this is helix A, helix B, helix C, helix D, helix E, helix F, helix G, helix H. So there's a bunch of those that make up this overall protein. Now, with that said, the polypeptide chains fold around a prosthetic group. That prosthetic group is known as heme right here. Heme is the prosthetic group. Now, that term, prosthetic group, is one that's important. Whenever I hear the term prosthetic group, I think of like a prosthesis, a prosthetic arm, a prosthetic leg, something like that. And I think, well, what does that do? That helps that person interact with their world. That helps them, pick something up or walk, something that their body might not have the capacity to do, which is why they have that prosthetic group or that prosthesis. Heme does the same sort of thing, but on a chemical level. And what I mean by that is that heme gives myoglobin, MB here, an ability to bind things that in its absence, so without that heme prosthetic group, this protein would not be able to do. Now, this right here, this is our hemoglobin tetramer. I want you to again look at, let's say, this A that I've circled. Or actually, no, not that one. I'm trying to find something that's most similar. This A that I've circled, can you give me a Y or an X? A Y if you see it, an X if you don't. Uncircle the other one. Well, you have that same similar structure. B helix, C helix, D helix, E helix, F helix, G helix, and H helix. The proteins are structurally very similar to one another. Now, one thing that has been misleading in the past, and I always say this, but myoglobin is a monomer. Hemoglobin is a tetramer. This is kind of application. Hemoglobin has a quaternary structure because it's made up of 4 different polypeptides. Myoglobin does not have a quaternary structure because it's only made up of 1 polypeptide. Next important detail about this is that hemoglobin, HB, does not equal 4 MB or myoglobin subunits. So don't think that, oh, well, if I just get 4 myoglobins to assemble, I'll make hemoglobin. That's not what's going to happen. But I want you to recognize that the structure of these 2 proteins here and here, they are very similar. And this structure is known as a globin fold. Next up, here's our kind of look specifically at myoglobin. Myoglobin, we've got our heme group, which binds O2. And this amino acid right here, that's kind of it's got like a light blue pigment to it. That helps stabilize and position the O2 molecule that we see right here. Approximately 90% of the protein is made up of alpha helices. And after that, heme, our individual or our prosthetic group, is a ring structure that looks like this. Now, we've got a histidine on the lower side, right here, histidine, iron in the center of that heme ring, O2, and then another histidine. So it's histidine residues. Remember that kind of five-membered ring structure that helps stabilize this O2 above this ring. Now, an important thing to know about this, Fe2 plus. Well, iron is an important mineral that you get in your diet. Um, in the absence of iron, I know that we talked about this the other day, someone might develop the affliction known as anemia. Well, what happens when you are anemic? You don't have a lot of iron in your blood, but also you get tired. And that's in part because your body can't shuttle oxygen around, which leads to a decreased metabolism, which leads, or sorry, that decreased metabolism is you're not going to be able to do as much of that oxidation of sugars from, or that involves O2. So, in the event that you have a low iron content or you have the wrong iron ion, we need Fe2 plus. If that Fe2 plus is oxidized to Fe3 plus, it's not going to bind to O2. Now, when this happens, here's what I want you to recognize. We've got about 3 more slides, and then I'm going to say that's the end of our stuff for this exam. Okay. This structure right here, that is a kind of side angle view of our heme ring that has not bound oxygen. Can you give me a 1 in the chat if you can see how it has not bound oxygen? Okay. Now, on the right-hand side, that's our model that has bound oxygen. I want you to recognize a couple of things. And look at my hands to be like where my middle fingers are meeting. Imagine where my middle fingers are meeting is basically where that green iron ball is. When O2 binds, what happens is my middle fingers kind of shift up. So that iron ion, you could think about that iron ion basically being pulled. Now, looking at this model and this model, our Y and X models, does it look like the length of this bond changes at all? Y or N to the chat? I mean, it's kind of tough. If you look closely, it shouldn't look like it changes because it doesn't. Now, does it look like this iron ball shifts, maybe shifts up? Yeah. And whenever that shifts up, what happens to the rest of this histidine ring? Go ahead and put N for no change. It goes U for up and D for down. Yeah, it shifts up. Okay. So when oxygen binds to that iron in the center of our heme ring, it kind of pulls this iron ion up. And that iron ion pulls everything else below it up. Okay. We have a movement of 0.4 angstroms. It's a very, very small distance. But the significance of this is, has anyone ever heard of the butterfly effect? Add a Y or an N to the chat. Okay. So butterfly effect is an idea of a butterfly flaps its wings in Tokyo, which leads to something else happening across the other side of the world because it's a chain of events. We have this sort of chain of events. O2 binds, pulling the iron ion, shifts the iron ion up, which causes this heme ring to shift up, which causes everything here to shift up. Okay, well, That histidine, that histidine ring is a part of the protein. And so what we see is, yeah, it's the iron ion moving, but it's also the entirety of the protein moving. That's well represented here with this blue cylinder versus this pink cylinder. The blue cylinder is basically showing an alpha helix in our polypeptide where it is initially, and then the pink helix shows where it is after it binds, after the oxygen binds to that heme ring, or the iron within that heme ring. Now, this O2 binding shifts or pulls the iron 2 plus in the porphyrin plane, that's the heme ring plane, or heme ring. It pulls it into the plane, and the distal histidine, the lower one, and its helix. These changes result in a shift in subunit arrangement. This is a huge impact because it takes a protein or takes hemoglobin from its T state to its R state. Its T state known as deoxy state, and its R state to its oxy state, or its oxygenated and functional. Now, this is kind of another perspective of that. So this is hemoglobin that has not bound to any oxygen. And now watch closely. That's hemoglobin that has bound to oxygen. Give me a response ranging from 1 to 3. 1 being, oh my gosh, that was fascinating. And 3 to, I didn't see anything. Okay. For those of you who said 1, awesome. I remember seeing this and being like, and not this specific one, but similar models. Okay. What is really helpful for me is to see, to like draw my attention to something specifically. I'm going to draw your attention first to our heme ring, where the oxygen is binding. So what we're expecting to have happen is oxygen is going to bind here, and that is going to pull this alpha helix kind of in this direction. Can you give me a Y if you follow that, and N if you're like, okay, no, I don't believe you. Okay, so it's going to happen there, but then we've got the same thing happening here, here, and here. Because add to the chat, how many O2 molecules can hemoglobin bind? 4. Hemoglobin can bind 4 because it has 4 heme rings, 4 places to bind O2. So we've got this hemoglobin that's going to bind, or this heme ring that's going to bind to O2, which is going to cause this shift, and then another one, which is going to cause a shift, shift, shift. Can you give me a Q in the chat if you follow that, or a V in the chat if you... Wait, could you say that again? Okay. So the idea is that in all of these places where I'm putting big circles, oxygen is going to bind and it's going to cause these alpha helices here, here, and then one back here and one back here to move. So this binding is going to cause all of these different movements. Now, I want to erase that because now what I'd like to do is draw your attention to something a little bit more, hopefully dramatic. There's a central cavity in this protein. Put a box around that central cavity. And you know what I'm going to do actually is I'm going to use a different tool to make it a little bit more kind of stationary. So I'm going to use the annotate tool, and I'm going to draw boundaries of this central cavity. Okay. Oh, that's not the one. Whenever I switch from this slide to the next slide, these two images are basically perfectly positioned so that I'm going to see an overlay, or a transition is going to give me an overlay. So there's my central cavity. That central cavity has basically got four sides to it. That central cavity is in that size. You don't have to remember like how large it is or anything like that. But what I want you to do is visualize that and say, okay, that's how large that central cavity is when my protein is deoxygenated. That region in that center, it does oxygen doesn't bind there, but when we go to the Nick state, the R state, the oxygenated form of our protein, which that oxygen binding has taken place distant from there, let's see how that central cavity changes. Okay, what happened to that central cavity? Did it stay the same size? Y or N to the chat? No, it didn't. It got smaller. Now, whenever I toggle back and forth between these structures, after I look at that central cavity, some of the other parts of this protein changing or not changing become a little bit more clear to me. For instance, I'm going to circle something and I want you to recognize it. Okay, so can you give me a one in the chat if you see that one that I just put a circle around? Okay, and then I'm going to try it over here. I'm going to circle one over here. That one corresponds to the N termini, so the first amino acid in the structure of one of these or of these different subunits. So I've circled two of those N-terminal peptides. Now, looking at that N-termini, if I jump ahead, oh, it moved. That means the N-termini of my protein moved. And if you look at this N-termini from its T state or its deoxygenated state to its R state, the protein has kind of like recoiled almost. So what I want you to take away from this right here, and this is going to be the end of new material or the material that's going to be tested on this first exam. What I want you to take away from this is the fact that we have a protein that will bind oxygen, and it's going to bind it in four different locations. But this oxygen binding, well, it's taking place in different locations, but then we're seeing how this oxygen binding event causes changes in the structure of the protein. That's an important part about quaternary structure, about proteins and how they function. This binding takes place in one location, and the structure of the protein changes in a completely different location. Just making sure the exam is next Thursday, the 25th. Yes, that's correct. Okay, I'm going to stop right there. I'm going to stop my recording. If anyone would like to hang around and ask any questions,